In this video, we're going to talk about reciprocal regulation and cascades. These are the last two major patterns of metabolism I want to cover before we start talking about individual pathways, so it's cool that we're getting that close. I'm going to illustrate this using the example of glucose and glycogen, which we covered in our video looking at homeostasis and sugar metabolism. So if you haven't seen that one yet, you might want to check that out first. I'm also going to use the SBGN, the systems biology graph notation, a lot. So if you haven't watched that video, you might want to check that out before digging into this. But hopefully you're prepared and ready to go, and let's launch right into this, talking about reciprocal regulation and cascades. So the stars of our show are going to be glucose and glycogen. This is the free energy form, not free energy of like a delta G, but this is available to do stuff in cells to be used as energy. And then glycogen is just the storage form of glucose. So basically, it is an array of these six-membered rings that are glued together. The way that this is built is we just stick another glucose on, and the way that we free a glucose is we just chop it off so it can become a glucose. And what I'm going to do is talk about this glucose-glycogen system, and we're going to come across this notion of both reciprocal regulation and cascades sort of in situ. So we'll expose it as we go. But for now, let's just think about this as a cool biology issue. So what we're going to be thinking about is inside of a hepatocyte, inside of a liver cell, the problem that's faced with energy storage, right? So it can keep things as this glucose form here, which is free and accessible, or it can store glucose to use for later. And so there's this possible mechanism, these reactions, right? We can go from G1P, that's the glucose form that becomes glycogen, or we can go from glycogen into G1P. Both processes are possible. Both are catalyzed by enzymes. So we have PYG, which is a phosphorylase that takes glycogen and breaks it into G1P. And then we have synthase, GYS, which takes G1P and turns it into glycogen. And the question our cells face is at any given time, which of these should be on? And as I've drawn them right now, they're kind of both on, right? And that would be kind of a bad situation, right? Because we're breaking down glycogen and we're forming glycogen. And to the extent these are both running at a high velocity at the same time, you're basically just spinning your wheels as a cell. So what we really want is a way to basically favor doing one over the other, to favor either breakdown or favor formation, but not both at the same time. The way that this is achieved is using phosphorylation. So you might notice here that there's a little P at the edge of this. In systems biology graph notation, that says this is a phosphorylated enzyme. And indeed, we can draw this over here that we have PYG is in an unphosphorylated form here on the left, phosphorylated form here on the right. The phosphorylated form can catalyze this reaction. The unphosphorylated form can't. What that means is effectively this is off and this is on. So when it is phosphorylated, PYG goes on. What that means is you start breaking down glycogen. With GYS, if you phosphorylate it, it now goes over here. It can no longer catalyze its reaction. So now it becomes off over here and on over here. So for GYS, if it is dephosphorylated, it is on. And if it's phosphorylated, it's off. And hopefully what you can see with these colors is there's kind of a switch, right? If I'm in the orange state, I am basically I'm synthesizing glycogen from glucose. And if I'm in the purple state, I'm doing the opposite. I am now breaking down glycogen, and I am not synthesizing glucose, right? So think about these as a correlated switch. The orange thing flips over to the purple thing. The purple thing flips over to the orange thing. That's a change in the state of the system. All right, cool. Now, I'm using this phosphorylation thing. Just a quick reminder of what this means. It means we're sticking a PO4 minus onto particular residues, typically a serine or a threonine, sometimes a tyrosine. But when we phosphorylate an enzyme, it can dramatically change its activity. And so this is an example of a different enzyme than the ones we're talking about, but it illustrates the point so well I kept it. This is a set of phosphorylation sites in orange that are buried when they're unphosphorylated, and they're exposed when they're phosphorylated. So imagine you've taken a bunch of negative charges and you stick them into that buried form, super unfavorable, right? Negative charges buried. Now you expose those charges. So when you phosphorylate it, it drives this conformational changes. It causes this big twist in the enzyme. What does that big twist in the enzyme do? Well, it takes this active site in blue from being buried to now being accessible and exposed. So the phosphorylation is literally changing the structure, changing the conformation of the enzyme, taking it in this case from being off and closed when it's dephosphorylated to on and open when it is phosphorylated. So we're seeing similar sorts of things happen in this system where phosphorylation is taking the system from being off to on or from on to off. All right, cool. How is this done? Well, it turns out that both of these enzymes are phosphorylated by the same enzyme called AMPK. And what AMPK does is it phosphorylates PYG, tipping it into the active form, and it phosphorylates GYS, turning it into the inactive form. So this is coordinated regulation, right? With one enzyme, we're able, if this enzyme is on, it's going to turn this on and turn this off. What turns that enzyme on? It turns out it gets turned on by AMP, which is low cell energy. If your cellular energy is low, AMP starts to build up. As AMP concentration starts to build up, it activates AMPK, AMPK. As AMP kinase activates, it activates PYG, which causes glycogen to get turned into G1P. And it deactivates GYS, puts it over into this form, so we no longer take G1P and make it into glycogen. So this one enzyme can reciprocally regulate these two sides, right? And just to finish this off, you almost always have switches like this that they can be turned on and off. There's the other side. So here I'm showing this AMPK turning it on, or turning on breakdown of glycogens. AMPK triggers breakdown of glycogen. PP1 does the opposite. It drives these reactions in this direction. It causes GYS to turn on and PYG to turn off. How is it activated? It's a little bit more complicated than AMPK, but it turns out at the end of the day, it's activated by high glucose. So if your glucose concentration goes up, that says turn off PYG, turn on GYS, start synthesizing glycogen. All right. So, and just for funsies, because biology is super cool and complicated, I'm not showing nearly all the arrows on these systems. But one other fun thing that PP1 can do is it can actually inactivate AMPK too. So it not only is, as your glucose starts going up, it says also turn off this system. So it's multiple sets of switches that all tip it towards we're going to take glycogen and turn glucose and turn it into glycogen. Okay, so this illustrates the principle of reciprocal regulation, where the forward and reverse pathways are controlled by the same signal with inverted logic, right? If I phosphorylate this system, it has opposite effects on synthesis and breakdown. If I dephosphorylate it, it has opposite effects on synthesis and breakdown, right? So as a whole, this system is reciprocally regulated. Okay. So that is at the level of, say, an individual cell. How do we tie this into hormone signaling, right? And recall in the last video, high-level video of this, I said if you have a cell, a muscle cell that's consuming glucose, that's drawing glucose out of the blood, your islet cells start dumping out start dumping out glucagon. I'm sorry, all of these names are so similar. I have a theory that biochemists actually hate everybody and come up with very similar names just to be annoying. But glucagon, so islet cells dump out glucagon, that causes liver cells to dump out glycogen. And if the opposite's true, you have high blood sugar. What you start seeing is glucose gets imported and stored as glycogen. And what we're going to be thinking about for the rest of this video is just this switch right here. Sorry, not that switch. Circling the wrong thing. This switch here, when glucagon is high, glycogen gets made into glucose. When insulin is high, glucose gets made into glycogen. How is that switching achieved? And with this, we're going to illustrate a reciprocal regulation and cascades. Okay, so here's the switch that I already showed you. It's gotten smaller, and what that means is it's about to get really complicated. But the pattern is so cool, hopefully, that you can appreciate it even with the complexity. So what I'm showing here is, again, the switch. PYG is in the on form. It's causing glycogen to break down. And GYS, when it's in the when it's unphosphorylated, it's causing glycogen formation. So phosphorylation switches us to break down glycogen. Dephosphorylation switches us to break down or to form glycogen. Okay, cool. What we're going to try to do is link this at the top to glucagon, which is saying low blood sugar, and insulin, which is saying high blood sugar, right? How in the world do these signals, which are up in your bloodstream, get down to this individual switch? So the way I'm going to do this is I'm actually going to build up from the bottom. So from this switch, and we're going to do some sort of zooming in and out here. But if we look at this switch, what we see is PYG is phosphorylated to be made active. It is phosphorylated by another enzyme called PHK. So PHK takes an ATP, takes a phosphate off of it, sticks it onto PYG. PHK, it turns out, is also in the same form, that it is phosphorylated, it phosphorylates PYG. Okay? So if this is phosphorylated, glycogen gets broken down. Okay. At the next level, we added yet another level. In this case, it's this molecule pKa. And what pKa does when it's active is it phosphorylates PHK. So if PHK is active, if pKa is active, it phosphorylates pKa. I can do this. There are so many letters because remember, biochemists hate everybody and want your brains to explode. So the names are just all the same. Look at the picture, and then if I say words that don't match the picture's right. pKa, when it's active, activates PHK. When PHK is active, it activates PYG. When PYG is active, it activates glycogen breakdown. Okay. pKa also phosphorylates GYS over here. And remember, when GYS is phosphorylated, it goes into the off form. And so when pKa is active, it turns off glycogen synthesis. So pKa is one molecule that can both activate glycogen breakdown and inhibit glycogen formation. All right. Now, this hopefully looks familiar because we just did it with AmpK. Remember, AmpK does the same thing. AmpK is working directly on PYG and GYS. With pKa, we're seeing the same pattern, but we now have this indirect step. We go through this PHK thing in the middle. Okay. We stretch. We're going to make another layer. Turns out pKa is activated by this molecule cyclic AMP. This is a small molecule that's actually a derivative of ATP. So ATP gets turned into cyclic AMP. Cyclic AMP turns on pKa. Where does cyclic AMP come from? Well, cyclic AMP comes from an enzyme, again zooming in, called adenylate cyclase, which goes from an inactive form to an active form. And what causes adenylate cyclase to activate? Well, oops, wrong way. It turns out it's this protein called G-alpha. And what causes G-alpha to activate? It turns out it's the active glucagon receptor. In fact, if you watch my videos about allostery, we talked about how G proteins are activated by GPCRs, G protein coupled receptors. So the glucagon receptor is a GPCR that when it binds to glucagon, activates, which activates G-alpha, which activates adenylate cyclase, which causes adenylate cyclase to make a lot of cyclic AMP, which activates pKa, which activates PHK, which activates GPYG, which finally causes the breakdown of glycogen on this side. Simultaneously, pKa inhibits the synthesis of glycogen. What we are seeing here is a cascade, right? Where one event up here triggers a stack of further downstream events, ultimately leading to this interior outcome of glycogen getting broken down. All right, now we're going to talk about why cells use cascades in a little bit. Before we get there, though, let's fill in the other arm. What in the world happens with insulin? So it turns out we're going to start just like we did with glucagon and build up. We're going to build up from this internal switch. So it turns out the first step in this switch is one we've already seen before, PP1. Remember, that was the phosphatase that that basically puts PYG into the inactive form, disabling glycogen breakdown, and puts GYS into the active form, turning on glycogen synthesis. Sorry, zooming out here. And what you'll notice is this PP1 complex over here is able to activate this and this, and it actually also activates PHK. So that was the enzyme that activated PYG. So this one enzyme, PP1, drives the system into a state where it's going to prefer to synthesize glycogen. Okay? And we're going to discard. So we're going to come back into our slides, zooming out. Okay, we're back. And we've seen this before, right? So this is PP1. Now it's in a slightly different form, but PP1 is doing the same basic activity. Okay, so PP1 flips a switch to prefer this. How is it activated? Well, it turns out it is activated by a molecule conveniently named pKa-B. So pKa-B activates PP1, which then turns on the synthesis. What activates pKb? Turns out pKb is activated by this molecule, PIP3, which is actually a phosphorylated lipid. So it's a small molecule, just like cyclic AMP. What activates PIP3, or what makes PIP3? Turns out it is activated by this molecule called PI3 kinase. So PI3 kinase starts making a lot of PIP3. That turns on pKab. That turns on PP1. That turns on GYS, turns off PYG, makes us synthesize glycogen. Oh, we are so almost done with this. Last step in the middle there is what activates that? Well, it turns out insulin does. So insulin in the bloodstream activates the insulin receptor. The insulin receptor actually phosphorylates the insulin receptor substrate. This is a kinase that this isn't a kinase, but this activates PI3 kinase through an allosteric mechanism, which then leads to the rest of this process, right? So what we have here is a set of signals: active, 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 build up something, active, active, and then that shifts this molecule to active, this molecule to inactive, this molecule to inactive. The net result being that an insulin signal in the bloodstream causes your glycogen to be synthesized and not broken down. Again, this is an example of a cascade. Signaling cascade, right? What's so cool about these? And there's a couple of things that are cool about them, but one of them is it amplifies the signal, right? So one signal input here can get multiplied over and over. So you can get more than one G-alpha protein can actually be activated by a glucagon receptor. You can get many, many cyclic AMP molecules synthesized for every G-alpha protein, right? This is turning on an enzyme. The same thing goes here. These kinases, right? These are just using ATP to stick phosphates on things. So every time you do this, you can get an amplification. So you can get, a 10,000x amplification for one glucagon molecule. You can get 10,000-fold amplification of PYG activity. So it amplifies the signal. The other reason that these cascades are really important is they provide opportunities for crosstalk. So for example, cyclic AMP is made by many different processes. Things like PIP3 are made by different processes. Different enzymes can be kinases and can phosphorylate these subunits. Different enzymes can be phosphorylases to chop these off. And so what this means is this cascade actually represents a vote from glucagon. Glucagon is voting and saying, hey, guess what? I think we should start breaking down glycogen. Other processes are voting and saying, yeah, I really think we should break down glycogen, or saying, no, I don't think we should break down glycogen based on whatever metabolic state. But the net result is that we end up with a coordinated activity where, based on a particular physiological state, we get a particular outcome in terms of, in this case, glycogen breakdown or synthesis. But it's all controlled by these two interacting features of reciprocal regulation, where enzymes are activating and inhibiting opposing pathways. They're doing it through a signal like phosphorylation that has opposite effect on opposing enzymes. So PYG and GYS, phosphorylation, has the opposite effect on their activities. That lets the cell decide whether it's going to break down or form glycogen. And finally, regulation at physiological scales like hormones can be linked to regulation at cellular scales by things like these signaling cascades. Okay. So that's all I have on that. Hopefully it was an interesting and cool look at these two phenomena of reciprocal regulation and cascades. Also some really cool biology, right? I think it blows my mind, honestly, that a liver cell can sit there and can measure, well, that's the concentration of glucagon in the blood, and then decide what to do with its sugar stores based on these pathways, and vice versa for insulin. What we're going to be doing in future videos is unpacking each of these individually. Like, for example, we have a whole video on what is glycogen, and a whole video on the different other sorts of related metabolic pathways. Really look forward to seeing you in those. So anyway, thanks again for your time here, and I will talk to you later.